have the right conditions to live, and then they collapsed and they eventually became extinct. So here, the interesting analysis is that here, the Columbian mammals from the basin of Mexico, it seems that the population is stable throughout the last 40,000 years before, I don't know what's happening. Is that me? Okay. Right. So this will be an indication that climate change didn't affect the mammoths in the basin of Mexico. But this is just a mitochondrial genome, so we need more data from the nuclear genome to have like more certainty to infer that information to make that claim. Another interesting thing is that they don't only have a stable population size, but also they have around 1 order of magnitude less of the woolly mammoths in Siberia. So this is telling us that the population of woolly mammoths in Siberia was around, the effective population size was around 10,000 individuals, something like that. And here in the basin of Mexico was only 1,000 individuals. So it was less diversity in Mexico City. I told you that there were 61 mitochondrial genomes. So that means that aside from the 28 high coverage genomes that we have, we also recover 33 mitochondrial genomes that were between 1x and 10x. And we perform an analysis that is a phylogenetic placement, in which, given the phylogenetic tree that we already have from the mammoths, we just assess what was the probability that one sample was placed within one clade or the other. And in that way, we assess that most of the samples belong to the subclade 1G1. So that's 14, and then 9 to 1G2, and 3 to 1G3. So the interesting thing here is that we can harvest information from all of the mitochondrial genomes that we have, even though we define the tree with just the high coverage ones. And this is also an interesting thing, is that we were able to sex the mammoths. So we were able to say which were females and males. Out of the 77 that I told you, we were able to accurately infer the molecular sex from 25 of them, and we observed that it was a similar ratio of males to females in the basin of Mexico. And this is interesting because a previous study suggested that the woolly mammoths in Siberia, there were probably more males than females. And it has been extrapolated, or it has been explained by the fact that the mammoths have a matriarchal society. So after the mammoths, elephants, but also like present-day elephants, and probably mammoths in the past as well, after the elephants get to a certain age, then they are expelled from the group, and they are wandering around by themselves, and this probably is more easier that they fall into a natural trap and die there. Or if they would have stayed with the group, then the elephants help each other. So the interesting thing here is that we are finding equal rates of females to males in the basin of Mexico, and that might mean that like the herds, but also lonely males came towards the lake, and they died there. So out of the inference that we also performed with nuclear data, we generated these genomes with this coverage. It's not really high, aside from this individual, and we compare this to reference mammoths from woolly mammoths here in Eurasia and steppe mammoths here in orange. And basically what we observe is that using this analysis, that it's not really important right now to explain what is the underlying basis, but it's basically if we have more genetic affinity to a certain sample, then it means that it shares a similar demographic history, and it's more closely related to that. So here we have the woolly mammoths. This is at the nuclear level. So here we have the woolly mammoth, the Columbian mammoth from Wyoming, and then the best Columbian mammoth from the basin of Mexico. And we're observing how it is associated to the different samples that I told you. So in green, we have other Columbian mammoths, in blue, the woolly mammoths, and in orange, the steppe mammoths. And we can see how the Columbian mammoths behave as a clade, So here, the Colombian mammoth from Wyoming is more similar to the Mexican samples. And then here, this sample, the best sample from Mexico, is more similar to the Colombian mammoth from the USA, but also to the Mexican samples. So another thing that we wanted to assess is that whether the Colombian mammoth from Mexico had the same amount of woolly mammoth alleles that the Colombian mammoth from the USA, and that means that is because I told you before that the frontier between USA and Canada was a hybridization zone. So there has been reports that the Colombian mammoth from Wyoming received an extra flow of woolly mammoth ancestry later on in time, aside from the hybrid origin. And this is what we observe here. So we performed this test in which each of the woolly mammoths that we see here, we wanted to assess if they were more similar to the Colombian mammoth of the Mexican sample. If the result of this test is significantly negative, that means they were more similar to the woolly mammoth from Wyoming than to the Mexican sample. And this is what we are observing. So a higher genetic affinity of whatever woolly mammoths that we are assessing here from the map that I show you from different geographical areas than the Mexican sample. And then another thing that we wanted to assess is that I told you that the Colombian mammoth from Wyoming had an important amount, 40% of their alleles were from a Krestovka origin, which is this steppe mammoth, really old lineage. And here the interesting thing is that, again, we have here any woolly mammoth sample, and here what we wanted to assess is that whether Krestovka is more similar to the Colombian mammoth from the USA or Canada than to the woolly mammoth. So you can see here that in all of the cases, samples are the result of this test is negative. What we want to compare is the result for the Colombian mammoth from Wyoming here in blue, than the Mexican sample here in green. And what we observe is that at least in two cases here, it seems that the sample from Mexico has more Krestovka alleles than the one from the USA here and here. That might be explained by the fact that since the Colombian mammoth from the USA received extra gene flow from the woolly mammoth and the Mexican samples didn't, then that's why you would find more of these Asian Krestovka alleles in the Mexican samples. And given that, I mean, we perform a phylogenetic tree using this nuclear data, and what we observe is that here we have Asian present day elephants from Asia, present day elephants from Africa. Here we have woolly mammoths. Here we have the Krestovka, sorry, the Colombian mammoths. So we see that the Colombian mammoth from Mexico is more closely genetically to the Krestovka sample than to the woolly individual. So that correlates with what I just told you about that we obtained from the F statistics. So one analysis that we want to perform now is that we want to take this genome from the Colombian mammoth from Wyoming, take out the part of the ancestry that we know that is from woolly mammoth origin, and then do the phylogenetic analysis again and see how it relates to the best sample that we have here from Mexico. So far, just a take-home message from what I just told you about. We are super excited that for the first time ever, we're able to recover DNA from Colombian mammoths out of tropical latitudes. We recovered 61 mitochondrial genomes, 28 of those at high coverage, 3 partial nuclear genomes. We observed that there were 3 deeply divergent lineages in the basin of Mexico, which might have been contemporaneous to each other. This might be an explanation of why we have differences between the Colombian mammoths from the USA and Mexico. And the explanation would be that when the hybridization process was happening between the woolly mammoth and the Krestovka, there was genetic structure, population structure in that woolly mammoth population. And they would have created different mitochondrial genomes in that. So after they had mixed, then that structure would have go into the Americas, and that's why we see that the mitochondrial genomes of the Colombian mammoth from the USA and Mexico are different. Another explanation could be that there were different flows, waves of a mixture between Krestovka and the woolly mammoth, and then we have different waves in time. That's the all mitochondrial analysis could be explained by that. And then at the moment, at the nuclear genome, the evolutionary history between the Columbian mammoths between the USA and Canada, they seem quite similar, aside from the fact that the Columbian mammoths from Mexico had slightly more Krestovka alleles. Just briefly, how much time do I have? 50? Okay, great. So I can take my time a little bit to explain this part. How many of you know here that camels are originally from America? Nice. We have eager students, that's great. So all the camels that you see nowadays in the world, the ones that you see in Africa, in Arabia, originally they came here from America. The oldest sample of camels is dated to 3.5 million years ago from Canada. And aside from that observation, also when you perform a phylogenetic tree with mitochondrial genomes, incorporating, for instance, guanacos, llamas, which are camelids, and camels from Africa and Arabia, but also ancient camels from Yukon in North America, you observe that genetic variation of the ancient camels, and these are camels from 50,000 years, is very closely related. It behaves as a sister clade to the genetic variation of present-day camels from across Western Eurasia and Africa. So they are not closely related to guanacos and llamas. They're closely related to camels that nowadays they exist in other parts of the world. And this makes sense because camels are actually adapted to really dry environments, regardless whether they are cold or warm. So if you think about camels that they live nowadays in Mongolia, during the day it's really hot, but during the night it's super cold. So it makes sense that camels originated in North America, where the climatic conditions were quite dry, and from there they colonized different latitudes and ecosystems where they adapted to them. So we were able to recover 3 mitochondrial genomes from camels from Mexico City, and with this data, 2 of them had high genome depth of coverage, and 1 of them had 3x coverage. And with this data, we wanted to assess how they related to the other ancient camels that had been previously recovered from Canada and dated to around 50,000 years. So, sorry about this. Shouldn't be the second poster session unless you allow me to bring Eduardo later on today. But so this is phylogenetic tree that we recovered from when we incorporated the Mexican samples. And what we observe is quite simple. So here in squares, in black, we have the samples that are high coverage, the ones that are reliable. And then here in circles are the ones that have low coverage and that we place phylogenetically a posteriori into the tree that was already defined. And we can see here how the genetic variation of camels can be divided in 2 subclades. The first subclade is represented by samples from the 3 samples that I told you before from Yukon, Canada, dated to 50,000 years ago, and then 2 samples from Mexico, and then 1 really high coverage sample, good sample from the USA, and then the 2 low coverage samples from 1 from USA and 1 from Mexico. Now, the interesting thing is that according to the archaeological record, this sample is quite old. It's probably 100,000 years old. And what this data is telling us is that this genetic variation, we are obtaining it because it represents different temporal moments. So different migrations that happen, different camel migrations that happen in time. So this was probably a first migration that happened from the USA going to both to Mexico and maybe to Canada. And those lineages went extinct or less common. One of them made it all the way to Mexico, and there was another migration that happened, probably also happening from Canada, from the USA, over towards to Canada and again to Mexico around 50,000 years ago, or maybe slightly before than that. And that would explain why we see these 2 patterns of samples that are from Mexico that are related to Canadian samples, but also samples from Mexico that are related to USA samples, right? Horses, so horses are also originated from the Americas. At the family level, horses originated in the Americas around 50 million years ago, and they were not bigger than a dog at that moment. And they migrated to Eurasia, and they evolved the phenotypical characteristics that we know from horses nowadays. They came back to Americas around 3 million years ago, and there were not only different species of horses in the Americas, but different genera. So a lot of variation of horses in the America, and they became extinct. At the end of the Pleistocene, most of them became extinct. And by the time where the Europeans came, there were no more horses, no more megafauna, and actually they brought their horses back. So the horses that we see nowadays here in Mexico and other parts in the Americas were brought by the Europeans. They were not the horses that originally existed in prehistoric times. So here we can see, this is a previous study by Heisman and collaborators from 2015, where the different diversity of horses are represented here in this phylogenetic tree, also using mitochondrial data, and it correlates with the geographical location from where they came from with this color. So you can see here that in Central and North America, there were at least 2 different genera of horses. So Harrington Hippus francesi and also New World cavalling horses. So again, we wanted to assess where within this phylogenetic tree of mitochondrial data did the Mexican samples fall within. And again, we had 2 samples that they were high coverage and 1 that was low coverage. So we performed pretty much the same analysis as before. This is the thesis from Pablo Uribe Herrera, who is also doing his bachelor project with me. Very briefly, this is a phylogenetic tree using more data than before. And we have different clades here. So we have an outgroup, the ancestor, which is Hippidion. We have here another clade, Harrington Hippus francesi, that I told you before that existed in the Americas. Here we have, so this is an outgroup, and this is a sister clade to this genetic variation. These are cavalling horses. This is the clade of non-cavalling horses, so you can find here donkeys, zebras, and so forth. And here within, this is the genetic variation of cavalling horses, New World cavalling horses. So here you can find that the letter is really small, but if you trust me, here you'll find prehistoric horses that are both from Beringia, but also from North America, dated between 20,000 and 40,000 years ago. And also you see here present-day horses. So present-day horses from domesticated present-day horses. Now, the interesting thing is that or samples from Mexico City, one of them was basal to all of this genetic variation. So it's really divergent of all this genetic variation that has been previously reported. And the other sample, one of the high coverage samples, it seems that it is more closely related to a present-day domesticated horse. And after considering all the different features that I told you before, the deamination patterns and the fragmentation patterns, it seems that this is actually a domesticated horse. Because in Santa Lucia, there was a military base, and it seems that the army, they buried the horses there, and it got mixed in the stratigraphic layers. So, but we have another horse, which was low coverage sample, and after we performed the phylogenetic placement, we observed that this horse claded within the genetic variation of prehistoric horses from Beringia and North America, dated to around 20,000 years ago, 30,000 years ago. So this is super interesting because at just one archaeological site, we are observing 2 different mitochondrial lineages of horses present. So one that is super divergent to everything, another one that is really closely related to other horses, prehistoric horses in the Americas. What is interesting is that not only horses, camels, and mammoths seems to be quite divergent here in the Americas, but also other species of megafauna from Mexico, they seem to be very different from their counterparts in North America. So for instance, this is a study published in 2020, where they recovered mitochondrial genomes from the American mastodon, and they observed that a sample that they recovered from Mexico is basal to all of this genetic variation. So here in blue, here. And also another project that recovered ancient DNA from Pleistocene black bears. They observed that the sample from Mexico is basal, again, to the genetic variation of other black bears. So we might be observing here a pattern in which genetic diversification is occurring as the megafauna and other animals were moving and dispersing throughout the continent, particularly into tropical latitudes. And then when that happened, they adapted to different environments, and that created a lot of genetic variation. So this is one of the reasons if getting DNA out of a mammoth is not exciting to you, which I wouldn't understand why it wouldn't, but this is an excuse, a reason why it's super interesting to understand genetic diversity happening at the tropicals, at the tropics, because we are probably observing something so quite exciting evolutive events that happened before an adaptation, and here is a lot of genetic variation that has not been sampled. We also recovered DNA from a giant sloth. This is not as good quality as the horses, camels, and mammoths. We are in the process of generating more data. Fun fact about sloths is that sloths nowadays, they have 2 claws and 3 claws, and they live in trees. And one would have thought, okay, so prehistoric sloths, they were terrestrial, and they have different sizes. There was like the giant one, this one, it measured like 5 meters, 6 meters tall when they stood in 2 legs. So one would have thought that when they evolved into living into trees, like they evolve and do that like the same species. But actually, it seems that the sloths that were living on the ground, the terrestrial sloths, already had 2 claws and 3 claws. So that evolution happened at this, in a convergent fashion. So 2 different lineages of sloths, they figured out that it was easier for them to continue to exist if they went to the trees and continued there. And yeah, that's super interesting. That's what is called convergent evolution, when different lineages go through the same process in an independent fashion. And well, whenever we have data, we want to understand to which species of sloths does the samples from Mexico belong to. Our first analysis suggests that out of the 3 samples that we have analyzed, 2 of them are Mylodon darwini, and 1 of them is probably Jeffersoni here. And briefly, just to end, another thing that we want to do is that we want to obtain DNA out of sediments to better understand the past ecosystem, so do a paleoclimatic reconstruction of how the environment looked like 10,000 years ago, 20,000 years ago, and do that correlation that I was telling you before. So like reconstructing the ecosystem time, and also observing what was the human presence in the area through time, and then better understand why the megafauna became extinct. Was it the change in the environmental conditions? Was it because humans overhunted them? So in order to do that, from the archaeological site in Santa Lucia, before the airport was built, we took samples from sediments, from different depths, from different stratigraphic layers. We have 10 samples, like 3 different locations. 4, we have, out of 2 locations, we took 4 samples each, and then a 3rd location where we took another 2 samples. We're still working on this data. Hopefully I could come in the near future, or I can invite you to one of my talks in the near future to obtain results from this analysis. But basically what we want to do, and what I've been telling you and repeating over and over again, is to address what this cartoon is conveying, where we have here the time in the x-axis. So we have here older times and more recent times here. And then with these colors and these bars, we're representing the different ecosystems that were present. So for instance, here in this hypothetical condition, we had a lot of grass in the past. And then there were more forests. And then as human domesticated crops, then we see a lot of crops happening here, right? And then here we have on the upper part of this plot, we have human presence. So we can assess how many humans were present given the amount of lithic material that we observed. But also we could make predictions from genetic data. And the idea is to assess how common were humans in that area, and how their population demography expanded and behaved, and then correlate the effective population size, let's say genetic diversity of those individuals here on the y-axis to the left. And then we can trace a line on what was the evolutionary trajectory of the different animals. For instance, a bison, a hypothetical bison or a hypothetical deer, in which in this case the bison, it is highly affected by the change of grasslands to forest. It's not so affected by the human presence, and then it eventually became extinct because there's a lot of crops and it cannot feed itself. But for instance, here the deer, regardless whether there's a change in the environment or the human presence, it is having like a favorable evolution on that place. We want to do this analysis with the mammoths, with the camels, and the horses, and better understand why they became extinct. And that's pretty much it. So for the future, we want to generate more data. We want to perform capture experiment analysis of the nuclear genome to recover all genomes. We want to perform capture enrichment analysis from plants in the sediments to reconstruct the paleoclimatic characterization, and we will interpret this along from data from other references. And with that, I finished. I acknowledge the work of a lot of people that has been collaborated in this huge effort. Paleontologists from INA, colleagues from abroad, from Sweden, which had helped us with some particular analysis, and particularly people from LIG, from the International Laboratory for the Human Research Genome, from the paleogenomics group that have been working this for a long time, staff at LIG, students from my group that you can see here from the pictures, and INA for giving us permission to work on these samples. And thank you for your attention.
This is the small gift that we have part of the organizing committee and our patronage.
Thank you.
Is there any question? We remind you that we are going to be giving raffle tickets to those that ask questions. So is there any question? Please, if you are getting out of the classroom, we ask you to be quiet, please.
There's a question over there.
Yes. People, please, we ask you to be quiet.
A huge amount of work. I was wondering, and this is just a speculation, right? All that diverging data that you found with the Colombian mammoth, would it be possible that it points to the existence of an unknown species, and the Colombian mammoth was just having an introgression translate, and you haven't found these other species? Is it possible, or just?
Yeah, it is, and I mean that's probably what is happening here. That or could happen here is that it seems that the Colombian mammoth was a mix between Krestovka, that is something that we don't really fully understand, and the woolly mammoth that we understand. So if they were different, if there was a lot of genetic variation from this Krestovka lineage, when it mixed with the woolly mammoth, then it would give us different scenarios. And this could be what is happening. So that there were different populations of Krestovka that are mixed with woolly mammoth at different time points, and that's why we see all this genetic variation. But the interesting thing is that we see differences at the mitochondrial level and the nuclear level. At the mitochondrial level, they're quite different and divergent, but at the nuclear level, we don't have a lot of data, but from the data we have, they behave quite similarly. So what might be happening is that what I also said as an explanation in that there was structure in the population of the woolly mammoth. I didn't say that explicitly, but the mitochondrial genomes from the Colombian mammoths are from a woolly mammoth origin. So what probably happened is that female woolly mammoths are mixed with male Krestovka mammoths, and that's how the Colombian mammoth appeared. So if there was structure in that woolly mammoth population, so a lot of different genomes present in a similar area, then that would create divergent lineages So if there was structure, and then after they came to the Americas, there was an expansion, they would create divergent lineages at the mitochondrial level, but at the nuclear level, they will behave similarly.
Any more questions? Wait.
Sure.
Thank you very much for your participation, doctor. But my question is, from all the samples that you have been excavating there in IFA, there is a certain point in the decaying rate of the genome of the mammoths that made them seem be useless or not taken into consideration as part of the research.
Right, yeah. So DNA degrades, not only DNA, but molecules degrade with time. So there are studies that claim that they have been able to recover proteins from dinosaurs. So out of the scale of preservation in molecules, you have DNA, that's the one that degrades faster, then proteins, and then lipids. I mean, lipids are super well preserved. But going back to DNA. So DNA degrades, DNA is like ice cream. It degrades really badly in humid and tropical environments. So what we're observing here with the mammoths is that there's not a lot of preservation. The preservation is really bad, but we do have endogenous DNA, and that's why we chose the strategy to perform capture enrichment strategies. If we would have sequenced directly, we would have lost a lot of money, and it would have taken us a lot of time to recover a mitochondrial genome. So yes, there's a lot of degradation happening, but there's still data. If you know how to recover, there's still data. And I mean, that's why you were able to recover genetic data from 1 million year mammoth from Siberia, right? The Krestovka Naricha that I told you before. That is dated to 1.5 million years ago. That's a lot of time. A lot of species have gone extinct in that time. Neanderthal appears and then became extinct. Different species went out. But the reason why it's preserved is because it was deposited in the permafrost. And the permafrost is the freezer of the world. And then, like, people are concerned of like global climate change, and they are becoming warmer and warmer again. But scientists are super excited because a lot of fossils are being underneath in this process. Maybe also some viruses and other pathogens, but most of the mammoths that have been found in Siberia are being found because temperature is rising up, and then they're defrosting, and then parts of the earth that they were not able to drill before, now they are accessible.
Okay. Thank you.
Yeah, my pleasure.
So we talked about, well, this like talk basically was a lot of information. No? So I was curious, how long does it take to make like this research? No, you said that yeah, they found a lot of fossils in
Right.
the IFA, no, and that, well, it's basically new material to work with. And I was wondering, like, how long does it take to get to this level of already knowing of certainty,
Right.
no, about everything?
I mean, there are different ways to address your question. Assuming that one has all the money at the moment, then maybe 3 months or 5 months. But you don't have all the money. You have to apply for money to get grants and so forth. So when we were recovering the paleontological data at the beginning, the pandemic was happening. So we had to go and recover the remains with different safety protocols. It took a lot of time. And then we couldn't access the lab here in Querétaro because of the social distance restrictions, right? So assuming that's not happening and I have an infinite amount of money, then yeah, not so much time. So you need to first do a sequencing round to assess the preservation of the DNA. And after you do that, you make a decision how you want to go. If there's not a lot of preservation, then you do capture enrichment. If you have a lot of preservation and you're extra lucky, you probably want to buy a lottery ticket because that don't happen so often. And you just sequence again, and by yeah, another 3 months, you will have the data. We have all the pipeline to do the analysis already ongoing, and they're automatic and they're modular and so forth. So like it would have taken us 6 months to have everything from zero to this if everything would have gone smoothly.
Thanks.
Yeah.
So you mentioned something about recovering the DNA from tropical latitudes.
Mhm.
So how does this compare to Arctic specimens?
Arctic specimens. Yeah, I mean, the thing is that the DNA is much better preserved in Arctic experiments. Like imagine trying to eat something that has been outside here in Querétaro for 3 weeks in the parking lot and eating something that was in the freezer on the fridge. So the thing with the permafrost and the Arctic latitude, higher latitudes, is that molecules are better preserved. So I didn't explain this, but DNA is degraded because of three main reasons. The first one is that when an organism dies, all the different compartments of the cell, they are lost. So the enzymes that they were in the cytoplasm, they go into the nucleus and start degrading DNA. So the first big degradation is because of your own enzymes. Then the second degradation is because of microorganisms that colonize that organic material. So bacteria, fungi, everything, they start to eat the organic material in that dead mammoth or dead human or whatever. So that's a second important amount of degradation. And then the third one is UV light, is water. Water attacks the bonds of DNA and it fragments DNA through sorry, I cannot say it in English, but it breaks DNA. So what happens is that you have a frozen environment and dry environment. There's no water. The microorganisms are not active because in cold environments, they will not grow. And then probably after the death of the organism, if there was a lot of, a dry environment and cold, then the enzymes from your body will not be as active if that happened in a tropical environment. So that's the main reason why degradation happened faster here in warmer areas. But the reason why we were so successful to extract DNA from Mexico, that sounds very tropical, right? Is that Mexico has a lot of ecosystems, right? So we used to have a glacier up of Iztaccíhuatl. Nowadays it's not present, but the higher ground from the basin of Mexico is really cold. Have you ever been to Ecatepec or to the state of Mexico? It's dry and it's cold, right? So it's not the same as being in Acapulco. So that's probably the reason why it preserves so well. So after there was a lake, and then animals came towards the lake to eat, and then they died there, either because there was the basin of Mexico was a huge lake that expanded and contracted as climatic conditions change. But also it had a swampy surrounding. So animals came, they got stalked, and they probably died out of hunger or something else ate them, and then they stay there. So probably at the beginning, the DNA of the organic material degraded really fast because it was a lake. But afterwards, as climatic conditions changed, it probably dried up. And then after a certain time, the size of the lake got significantly reduced at the end of the Pleistocene. And then, the Europeans were the ones that they really drained the lake, right? But some parts of the lake were already gone before the Europeans came. Those probably the areas where the most of the better preservation is happening.
So from the table you showed of the amount of bones you collected and the amount of individuals, is there any particular reason why mammoths were the most prevalent?
That's a really good question. So it seems that's a question for a paleontologist. So I'm a bit on the spot right now, but the reason is that because it was a lake, so lakes there's like a gradient in the lake. I don't know, to be honest, I don't know how deep was the lake, but probably with wind and so like the surface of the lake was like whatever was falling on the surface of the lake was going towards the middle, regardless of the depth of the lake. So the bigger the animal, the more difficult for it to move towards the middle. And that's probably why we find elements that are longer and heavier than the ones that are smaller. That's the explanation that had been given. But at this site, there also have been found smaller animals as well, like birds, fish, some amphibians. So I mean, you see the composition of the whole lake, but out of megafauna, this could be the reason of why we're observing so many mammoths. Yeah.
For now, it will be posted for the goal. Thank you all for coming, and we'll see you later for talk 2:30.
Thank you.
Para cortar la...
Mencionó algo sobre el nuclear DNA,
Ujú.
los mamuts,
Ujú.
y que necesitan extraer